I think I'll start with saying that it's never a good thing to be talking towards the end of the day, especially when the bar is set so high. I want to give a review about mitochondria, mitochondrial diseases, their diagnosis, and possible treatment. So, by all means, I'm not an expert in mitochondria. I just had good fortune of working with some talented people while I was at Baylor. So, mitochondria are these reticulate structured organelles that we see in the cytoplasm of a cell. These are bacterial in origin. By that I mean mitochondria are actually bacteria at some point, and they had a symbiotic relationship with the eukaryote, and over a period of time, they just got absorbed by the eukaryote and became part of the eukaryote system. And so there are like 100 to 1,000 of mitochondria depending on the cell type or the tissue type. So as you can imagine, this number of mitochondria depends on the function of those tissues. For example, if we take skeletal muscle, cardiac muscle, brain, these are the areas where you find largest number of mitochondrial concentration. And skin, blood, and hair are the areas where there are very little mitochondria. And the mitochondria are dynamic and they are motile. I'll explain what I mean by this in a minute. So, usually when we think of mitochondria, we think about energy production. That's the main function of mitochondria, but apart from that, there are a few other functions that are equally important, like apoptosis and scavenging of reactive oxygen species, and mitochondria also takes part in the urea cycle, amino acid catabolism, and few other little lesser important ones are the pyrimidine biosynthesis, heme synthesis, cholesterol metabolism, etc. and etc. So, since the mitochondria is bacterial in origin, it has its own DNA, which is a circular molecule, and it also requires nuclear DNA for its function, structure, for several other things. So when we look at the mitochondrial DNA, it's a circular structure, about 17 kb in size, and it encodes for about 13 13 polypeptides and few tRNAs and rRNAs. So every mitochondria has about 5 to 10 copies of mitochondrial DNA. There is no concrete evidence of this. It's an assumption that every mitochondria has more than one circular DNA molecule in it. And so mitochondrial DNA doesn't have a chromatin structure, and it doesn't have a DNA repair mechanism. So which basically means they take quite a bit of abuse, and you see a lot of mutations and a lot of deletions in mitochondrial DNA. So the 13 polypeptides that the mitochondrial DNA encodes for, out of those 13, 7 goes to the electron transport chain complex 1 goes to complex 3 polypeptides goes to complex 4, and 2 goes to complex 5. Complex 2 is the only complex in the electron transport chain that is not encoded by mitochondrial DNA. So when oxidative phosphorylation takes place, electrons can enter through complex 1 or complex 2, and they go through all these complexes, and at the end you have ATP production. So you can say, so then what's the need for complex 2? And complex 2 is made up of 4 subunits, and all of these subunits are oncogenes. So you do need that. And I have like several diseases mentioned here. And the genes that are in red are the genes encoded by the mitochondrial DNA, and the ones in blue are the ones coming from nuclear DNA. So what kind of defects do we see in mitochondrial DNA? So it can have point mutations, rearrangements, deletions, and depletions. So depletions most of the time comes because of a mutation in the nuclear DNA. And coming to the nuclear DNA part, what kind of defects do we see because of mutations in the nuclear DNA? It can be a defect in the OXPHOS system, either as a structural defect or an enzymatic defect. It can be an assembly defect for the electron transport chain complex. You can have defects in the mitochondrial DNA synthesis or maintenance of mitochondrial DNA, or the nucleotide synthesis. And there can also be defects because of mutations in genes that take part in the stability of the mitochondria and also the communication between the two genomes. There are a few other issues that arise from nuclear mutations. I don't want to go through all those things, but I want to specifically concentrate on two things: the transport and fission and fusion. The rest of them, they are self-explanatory, so I'm not going to explain that. So transport, what I mean by that is, as I mentioned before, mitochondria are required for scavenging of reactive oxygen species. So in neurons, whenever they fire, there is a calcium flux that happens at the axonal junctions, and mitochondria need to be moved to those regions to scavenge the reactive oxygen species that are generated over there. If the mitochondria can't be moved to that region, then there will be reactive oxygen species accumulation, and that kills off that neuron. You don't want that to happen. So you want to move mitochondria from place to place, and you don't have enough mitochondria to have them present everywhere. So that's the important part of transport. And mitochondria are attached to this dynamine-like protein that forms the microtubules, and they move mitochondria from place to place. So any mutation in DNM1 can lead to accumulation of reactive oxygen species. So, and fission and fusion, mitochondria, since they are bacterial in origin, they divide by fission and fusion. So one way of for mitochondria to overcome the mutation load in the mitochondrial DNA is by fission and fusion. Whenever there is when mitochondria sense that the genome is have a higher mutation load, they proliferate to compensate for the increase in the number of mutations or depletions, some kind of insult to the mitochondrial genome. So if these genes, like FIS1, FIS2, if those genes have mutations and if they don't allow the mitochondria to proliferate and divide by fission and fusion, then again you have a problem. So what is a mitochondrial disease? It can be primary and it can be secondary. Most of the times what we see is actually a secondary disease. It's not the primary. The primary is the disease that is coming because of mutations in nuclear or mitochondrial DNA that is causing defects in structure or function of mitochondria. Anything that's coming because of glycosylation disorders or IEMs or rasopathies, etc., these are all classified as secondary mitochondrial disorders. So what happens when there is a mitochondrial disease? What are the consequences? These are basically consequences of loss of energy or energy failure. You don't have enough ATP to perform the cellular functions, and you have altered metabolic pathways to compensate for the loss of energy, and you have increased reactive oxygen species and lipid peroxidation, and at the end, when the cell senses that there is no hope, it just kills itself by apoptosis. So this is the most interesting slide. What is the clinical presentation? I'm sure Dr. Sharpen agrees with me. So any symptom, any organ, any time, any scenario, and the progression can be anything. It can progress pretty fast, or it can be in spurts. And the inheritance is also any kind of inheritance. So for example, if we take inheritance of mutations in the mitochondrial DNA, then it's definitely maternal. But if the inheritance of from the nuclear DNA, then it can be anything. So the incidence is about 1 in 3,000 to 5,000, and which is actually a little higher than some of the inborn errors of metabolism. So if the incidence is this high, is there a way for us to diagnose these disorders, especially when we are saying that it can be any symptom, any organ, any time, that kind of thing. So the red flag symptoms, the symptoms being equal, there are some symptoms that are more important than the others. The red flag symptoms is the name given by Dr. Sumit Parikh from Cleveland Clinic. I like that name, so I keep using it. So some of these symptoms are not by themselves. When you see a group of these symptoms, then you can be pretty confident that it is a mitochondrial disorder. The most important one neurologically is seeing a stroke that is non-vascular in distribution, and basal ganglia involvement are two of the most, kind of hallmark symptoms for mitochondrial disorders. And recurrent encephalopathy, epilepsies, they are not very specific. The one other specific one is the lactate peaking by MRS. And audiologically, you tend to see a bilateral sensory neural hearing loss, and muscular-wise, you see very severe hypotonia and rhabdomyolysis, and cardiac, you see cardiomyopathy, and few other red flag symptoms. Ophthalmic, you see optic atrophy, and GI is liver dysfunction, dysmotility, and few other non-specific ones. The most important one among that group is exercise intolerance. So at what point do we consider electron transport chain defect and not some other mitochondrial defect? So the first thing is when you have an unexplained association of symptoms. When you have a GI symptom and then you have a seizure, that's when you should start thinking about an electron transport chain defect. So in case of electron transport chain defects, the progression is going to be pretty fast. And these symptoms can persist and they can worsen. Most of the time that's the course, but there are instances where suddenly everything is fine. So what are the diagnostic tools we have? So far, there isn't a single silver bullet kind of test that is available to diagnose mitochondrial disorders. It's always going to be a bunch of tests. So there are some screening tests, and usually you screen for several multiple organs, and most of historically people depend on muscle biopsy for any kind of testing. Among the screening tests, the usual blood side tests, electrolytes, and that kind of thing. The one important test that most of the physicians who deal with mitochondrial disease do is the lactate pyruvate ratio, but this test has its issues, I would say. You have to have a pretty good, talented person doing this test, because you can't draw the blood with a tourniquet, and the patient should not be under stress. They should not wiggle when you are drawing blood. So, yeah. Then, if you see a ratio, high ratio, then you can say there is a mitochondrial disorder. And by urine organic acid analysis and amino acid analysis, you can have clues, but nothing is definitive. So these are the clinical evaluations that most of the patients undergo before anybody suspects a mitochondrial disorder. So we have all these tests. After all that, if everything comes up negative, can you say that there is nothing wrong? No, you can't. I mean, you still have the mitochondrial disorder lurking behind. So there are a few clues that we get from some of these testing. So one of them, few of them, from amino acids, if you see an elevated alanine, glycine, proline, it kind of clues you that there might be a mitochondrial disorder involved. And from urine organic analysis, you see ethyl malonate, 3-methyl glutathione. This is actually a pretty good marker for mitochondrial disorders. And you see a lot of dicarboxylic acid elevations. And from acyl carnitines, you see low free carnitine, and there are few acyl carnitine species that are elevated that kind of clues you in, but not definitive again. So at this point, you start thinking about a mitochondrial disorder. So what kind of test do you order? And can you recognize the phenotype, and can you order a specific test? Do you want to go looking for a point mutation in a single gene, or do you want to do a complete deletion duplication testing of mitochondrial DNA? You go looking for whole genome sequencing. It depends, it varies from physician to physician. Most of the time, in my experience, they go whole exome sequencing. And when you just order a mitochondrial DNA sequencing, if you find a mutation, what does that mean? So does that mean the patient has the mitochondrial disorder? You can't say that, because here you have to look at the heteroplasmy. And heteroplasmy plays a big role in deciding whether you have the disease or not. And at what point does the heteroplasmy of the mutation starts trending towards homoplasmy of mutation? So over a period of time, that can change. So these are the things that you have to keep in mind. If we sequence all of our mitochondrial DNAs, I can guarantee that every one of us will have a single mutation in the mitochondrial DNA. But does that mean we have the disease? No. It's just that you need to reach a threshold where that mutation actually makes a difference and causes the phenotype. And like I said, the muscle is the gold standard of testing for mitochondrial disorder. And using muscle, you look at histopathology, and you look at mitochondrial structure by electron microscopy. And muscle is the tissue that is used for all the electron transport chain, enzyme analysis, and polyrography is actually a test of where they look at the oxygen consumption by the mitochondria, and you can do the CoQ10 measurements and carnitine measurements. And again, here you can't really say that muscle is the only tissue type that you need to test. You have to look at other tissues like liver, because not all genes are expressed in muscle. So there are certain genes that are expressed only in liver, which cause pretty severe mitochondrial disorders. And when it comes to proteomics, these are mostly research type tests. No diagnostic lab offers them. It's the blue native gel electrophoresis. It's basically a native gel looking at the complex formation. So the electron transport chain, the 1 to 5, it looks at the formation of those complexes, and also these complexes, they form supracomplexes. Like, for example, 1 and 3 together, they form a supercomplex. So any disruption in this complex formation can also cause mitochondrial disorder. So that's what you'll be looking at using this technology. And genetics, you have all kinds of sequencing. So these are the diagnostic conundrums. So what tissue to select at what time. And most of these tests, they are very finicky. You can't have one lab do a particular test and then compare it with other lab. The overall result will be the same, but if you are looking at the numbers, they are going to vary quite a bit. And it's always impossible, near impossible to distinguish between a primary and a secondary defect. So, while I was working with the mitochondrial people at Baylor, we came up with this list of tools to analyze the patient fibroblasts and come up with a diagnosis for these patients. So these are most of the times these are the patients who are suspected of mitochondrial disorder, or some of them had a sequencing and they had a mutation in a mitochondrial related gene, but a BUS, not a pathogenic mutation. So we use this battery of testing to delineate the function of mitochondria in those patient fibroblasts. And together, we had pretty good success. But you can't depend on one particular test and do a diagnosis. So mitochondrial DNA quantification by qPCR, cellular respiration is where you are looking at oxygen consumption by the cells. And the electron transport chain activity assay, and the mitochondria have a double membrane, so they have this membrane potential. Whenever there is a mitochondrial dysfunction, that membrane potential is lost. So looking at that tells you if they are healthy or not. And you can look at the mitochondrial morphology under a microscope. Most of the times they are thread-like, nice looking structures. If they are round or if they are too big, that means there is something wrong with mitochondria. And finally, analysis of the ETC complex formation by blue native page. So what do we have at GGC in terms of mitochondrial testing? So I started working on the mitochondrial DNA quantification. So this test is almost ready. We can start using it in blood and fibroblast. We can use the same test in muscle and liver, but the validation is not done in those tissues for obvious reasons. You can't ask people for liver and muscle samples. So, I will be working on the electron transport chain enzyme assays soon. Hopefully those tests will be up and running by March or end of March. And what do we have planned for the future is the gene panel with the 16 nuclear genes that are responsible for mitochondrial DNA depletion and the mitochondrial DNA sequencing. So with that, if you want more information regarding mitochondria, this is the place to go. Mitosoft.org, it's the Mitochondrial Society. It's run by the leaders in the mitochondrial field. So it's a pretty good site to get some information about mitochondria. So that's about it. I'll take any questions.